Okay, hi everybody. Welcome to the Phi Tau Sigma 2025-2026 webinar series. So today I'm happy and privileged to get to introduce Dr. Achyut Adhikari. So Dr. Adhikari did both a bachelor's in technology and a master's in technology in food technology from Tribhuvan University in Nepal. And then he came to the US and did a PhD in food science with an emphasis on food safety and food microbiology at Washington State University in 2012. Dr. Adhikari is currently the interim director for the Food Innovation Institute, as well as an Adrienne Gravois Brazan professor and associate director in the School of Nutrition and Food Sciences at Louisiana State AgCenter. Dr. Adhikari has authored over 80 research and extension publications and presentations, including book chapters, manuscripts, and internationally invited talks. He's led numerous USDA-funded food safety programs, both in the US and globally, and he's received numerous awards recognizing his leadership in research excellence, as well as leadership in food safety, both from the LSU Ag Center Global Network Award and his distinguished professorship. We're really excited to hear about some of Dr. Adhikari's really awesome research in his talk titled Novel Approaches to Mitigate Food Safety Risks in Controlled Environment Agriculture Systems. So thank you very much for being willing to talk, Achyut, and I'll stop sharing and hand off the presentation to you.
Awesome. Thank you so much, Matt. Yes, presentation mode or presenter mode? Is it okay?
Yeah, I think we see it in presenter mode.
Or is it okay? What about now?
Oh, there you go. You're good.
Good. Awesome. Thank you so much. Okay, good afternoon, everyone. And I really appreciate you all joining this webinar to listen to some of the research that we are doing here in Louisiana State University. So today, I'm going to highlight some of the work that we are working on the controlled environment agriculture, some of the novel approaches to minimize the food safety risk. So what is controlled environment agriculture? It's not generally known as the CEA in short form, and the CEA refers to the production system that intentionally control several environment variables like light, temperature, humidity, nutrient concentration, water, and even like sometimes the carbon dioxide to optimize the plant growth or fungi growth. The CEA, like in chip production system, growers manipulates those several variables in order to increase the yield. As I mentioned, they manipulate the temperature, maintain appropriate temperature for production, and also humidity and carbon dioxide levels as necessary if there is required by the crops, lighting, the intensity of the light spectrum, the duration, and the root zone. That means like by the fertilizer with the water, the concentration, and then the pH of the fertilizer and oxygen. So several different variables are actually manipulated in order to maximize the yield and then maintain the quality of the crops in a controlled environment agriculture system. So there are several different classifications of controlled environment agriculture. You can classify in several different ways. There is like an environmental control and depends upon like a low control, the row crops, like low tunnels, and higher control, environmental control, like greenhouses. But if you classify controlled environment agriculture based upon the production method, basically it is classified on 4 different types. One is the aeroponic system, in which the roots are suspended, as showing in the picture, and there will be a mist of the nutrient that is periodically sprayed onto the roots to supply the water and then the nutrient to the plants. The another one is the hydroponic system. That is the most popular one, and then we are going to discuss about in more detail some of the food safety kind of like a novel approaches here in this presentation. So in hydroponic system, it's the most common method here in the system. We use like water soluble nutrient. And then water soluble nutrient is actually supplied to the crops. They absorb from the root and then grown in a controlled environment system where the temperature, humidity, light intensity, and several other variables are controlled. The another one is the aquaponic system. It's a symbiotic system combining aquaculture. That is like you can actually raise fish in the system, and then the poop of the fish, which is the nutrient for the plant. Okay? So those with the feed is the nutrient of the plant will be supplied to the production of the crop. So the popularity of aquaponics is growing really much also because of the fees and then also crop, and you don't have to purchase the nutrient for the crop. The fees provides nutrient for the crops. And the another one is the substrate-based fungal production. I normally see those into a mushroom farming system. But here we are going to discuss more about the hydroponic system. So what are hydroponic systems? If you see the definition from the USDA, okay, it's a cultivation technique where the plants are grown without the soil, and it uses a nutrient-rich water solution. And so in the system, water delivers all essential nutrients directly to the root zone, allowing the precise control over plant nutrition, water supply, and growing conditions. So there are several different benefits of hydroponic system. That's why its popularity has been increased so much. Even though, like it's a water-based system, you don't use soil. It's really interesting that, like it's very much efficient on utilizing water also. So 80 to 90% less water is used than the traditional soil-based system. Again, as I mentioned, controlled environment. So you can manipulate, like the different variables, temperature, humidity, depending upon the efficacy of the plant to grow, like maximize the yield, maintain the quality, higher yield than conventional method. If you compare the size or like the space that is utilized in hydroponic system, big turnover can reduce the land use, and then it's a low carbon print. That's why like so many popularities, that's so many benefits, and that's why the main reason this popularity is going really high. But there are also some challenges related to the hydroponic system. Like there are some concern, as we mentioned just now, it's connected through the water nutrient system. All that system is connected, and then it's in a controlled environment. As a microbiologist, food safety, many of you know, like there are different types of microorganisms, and especially those pathogens that are everywhere in the environment, and especially those environmental pathogens like Listeria monocytosis. Okay? They are always everywhere in the environment. And if they somehow contaminate the system, or come from water or through the seed, then it will contaminate the whole system. Or, like if something get contaminated, like water from fecal contaminants, like E. coli 157 salmonella, or if the seed get contaminated, if even only one cell or a few cell contamination, of course, that in the seed, the whole system is so much connected that there is so much high risk. Okay, and there is always a concern among growers is they don't really want to use the chemical sanitizers, because chemical sanitizers, the residues, they have other detrimental, like effect on the quality, and the consumer perceptions towards chemical sanitizers are not very positive. So this is a really big demand of like different techniques in order to minimize those microbial risk on hydroponic system. So we did this work in order to really understand what exactly going on, and how the pH of the nutrient solution, the composition of the nutrient solution will affect the growth of this environmental pathogen in Listeria monocytogenes. Okay. So with this work, you can see one of our results. There are three different figures. So one that designated by A is the growth of Listeria monocytogenes in distilled water. We use three different pH conditions: pH 5, pH 6, and pH 7. In y-axis, you can see the Listeria levels. And then in x-axis, you can see the time we observe up to 72 hours. And in the distilled water, we didn't see much changes. At lower PS, at PS5, there was a reduction, but in PS6 and 7, it kind of like stays kind of plateau. That's the same level. If you check the tomato nutrient solution, kind of like a very much similar pattern. We see a plateau, not much changes, and later after 48 hours, we see an increase in growth in PS7, but still that is not really high increase. But if you see the microbial growth in latest nutrient solution, all PS5, 6, 7, after 6 hours, we see a significant increase in the growth, up to 2 to 3 log increase in this latest nutrient solution. So nutrient solution can also affect the growth of potential pathogens. Here, also, you can see, we use 3 different varieties of the nutrient solution. It's similar to the previous chat, tomatoes, is kind of like a similar to dish control is with the distilled water without any nutrient. Okay, so it's kind of like a similar behavior, lower level, but in a strawberry and lettuce, even at all those 3 PS level 5, 6, and 7, there was a significantly higher level that we observed. That is within 72 hours. This one a little bit off, and then okay. So give me a minute. Okay, I wanna. Okay, great. So this chart shows the growth of Listeria monocytogenes and lettuce seeds and leaves. Those bar diagram. We wanna see, if there is a contaminated seed, how will it behave in a hydroponic system? Okay, how will it contaminate the nutrient solution or the plants or the leaves? So here with the bar, you see, we started with a contaminated seed up to 4.5 log, and the label actually remained kind of like a same throughout the production system. There was a decrease in 20 days, but the label remains same. At 8 days, we actually added the fertilizer solution in the hydroponic system. So this work was done in a biosafety second lab, and then it's a biosafety the chamber. We have a very small hydroponic system that we can actually use inside the biosafety cabinet. So with the use of that cabinet, the system, we added the nutrient solution on the 8th day. So you can see here all the way down, the graph that shows at 8 days, we didn't have any count on the fertilizer solution, but after we add the fertilizer solution on the 10th day, when it get connected with the root, the level of Listeria monocytogenes increased more than 1.5 log. And then gradually there we see the reduction in the levels after 25 days, but the root, it has kind of the similar level. It maintained that level, and then you can see the same thing with this bar chart, where the levels were almost between 3 to 4.5 log over the production period. So if the seed is contaminated, then it will contaminate the whole system. So we were, working on different ways to minimize those risks, and one of the best way that doesn't really leave any chemical residues is the UVC light sanitation technique. Okay, we have done a lot of work on UVC light, and we thought okay, let's use an UVC light and then see how will it affect those microbial risks in a hydroponic system. So we work with the seed treatment. We work UVC light exposure during hydroponic production. And then we also treated the UVC light, treated the leaves with the UVC light. So with this graph here, you can see, the Listeria monocytogenes contaminated seed, when it was exposed to UVC light treatment, the UV intensity was around 0.28 milliwatt per centimeter square. Within 40 minutes, the reduction was almost like 1.2 log. Okay, so within 20 minutes, we have a significant reduction that was around like 1 log in the seed. The 1 log reduction is approximately around 90% of the reduction. In many cases, if you have a contamination on the seed, you will not have a very huge microbial contamination, even though there are a few numbers. And if the UV light can reduce 1 log level within 20 minutes, that is a really great benefit. We also were concerned if the UV light will affect the germination properties of the leaf of the seeds. So we did the germination study, and it was an interesting results that actually after the UV light treatment, like you can see here, there was an increase in germination percentage of the seeds. It was not significant, not like an increase, but we did see increase, like in 20 minutes, 90% germination, and then in 40 minutes, that was like 95% germination. We also did the tetrazolium test, and like normally tetrazolium test detects the respiration activity, like in living tissues. Like because living tissues produce dehydrogenase enzyme, and then in this case, like triphenyl tetrazolium chloride, which is a dye that is used, and this dye will change if the living cells, like if it's a living cells, then it will change into red color, and if it is a dead cell, like it will have no color, or it will be on its stem. Okay, so with this test, it was like 95% of the viable test. We did this test because in many cases, like you may have a dormant seed which does not necessarily germinate very quickly, but may still be viable. Okay, so with the UV light treatment, it was actually not affecting the viability or the germination ability of the seeds. So this figure shows the effect of UV light on stem monocytogenes on different nutrients. We also want to see how the UV light direct application treatment with the fertilizer solution, like will that affect or reduce the Listeria levels on this fertilizer solution or not. It's really interesting results. We use with a very high level of contaminant, around like a 5.5 to 6 log contamination. And within 5 seconds, there was a significant reduction. And when it was exposed to 15 seconds, the reduction was like almost like 4 log and more than 4 log in many cases. So that was like a really good significant reduction. This was happening in all different nutrient conditions, like in strawberry, tomatoes, and even in lettuce nutrient solution. We were also interested on looking at like how the UV light treatment would work on lettuce surface, okay? Because like a lettuce leaves, it is not a smooth surface. UVC light is a surface sanitation technique, okay? Works well with the fertilizer solution, but when it comes with the green leafy vegetables, the surface matrix is very much different, okay? And we want to see if that has any effect. And we did inoculate the lettuce leaf and then expose to UV light. And you can see here, there was a reduction within 1 minute, which was not significant, but exposing up to 5 minutes, around like 300 seconds, we did receive a significant reduction, around 2 log of those reduction. And 2 log reduction on lettuce surface is a really great reduction. But again, like remember, this is for up to 5 minutes. But when you go up to the 5 minutes, like the treatment, there are like not positive results. Okay, so when you are identifying treatment procedure, you need to make sure that it minimizes the risk, but also maintain the quality of that product. Okay, so when we increase, when we did the treatment, UV light treatment, even at 1 minute, you can see those black spots at 5 minutes. There was like a lot of discoloration, probably with the effect of anthocyanins and several compounds. And that research does shows that if you increase the dose, or if you expose produce the UV light for a longer time, there will be phytotoxic. So we decided to go below 1 minute to maintain the quality of the produce also. So here, this graph, you can see we use two different types of lettuce leaves. One was more smoother in surface, and then the another one was more rough in surface, to make sure that how it affects. With a smoother leaf in 50 seconds, you can see there was a reduction more than 1 log, but in a rough surface, there was not much reduction. It was more like a plateau. Okay, so we didn't observe much reduction. And then if you see in that figure 2, we use this UVC light, that 50 second of a periodic exposure during hydroponic production system. So instead of just exposing for one time, like our idea was, if you are doing hydroponic crops, controlled environment agriculture, there may not be worker working in the night. In the night, you can have an expose of UV light for 50 seconds. That will, if you do that continuously every day, over the period of the time, that may control, microorganisms, or it will minimize, the spread of any contaminant that may get into your system. So here you can see with that 50 seconds of exposure, within 2 days, we got a significant reduction, and then increasing that, it was like plateau after 2 days, but you see a significant reduction within 10 days. That did not really happen similarly, just as opposed to when you just doing the control. So the major finding with these results is Listeria monocytogenes levels were significantly reduced on lettuce seeds after 20 minutes of UVC light treatment without adversely affecting the germination properties. The surface matrix of the lettuce leaf just affect. It's a smoother or a rough surface. And then our data shows that if you have a periodic exposed short time over the period of the time, it will reduce that is with this Listeria monocytogenes. We're also interested on looking at how the fertilizer contact surface will be affected with the UVC light, because there are several different types of materials that are used in hydroponic system. And then, if you have a contamination over the period of the time, it may form biofilm, and then continuously spreading those microorganisms in the system. Okay, so we use 3 different materials that we're using hydroponic system. Those coupons, we cut it into 1 centimeter square, and the metals that we use were actually used in a hydroponic system. Okay, so we use SDP coupons, PVC coupon, and vinyl coupons. It's around 1 centimeter square coupons. We use the control with the distilled water and then the fertilizer solution. We dip those contaminated coupons in those control and fertilizer solution, and then expose to UVC light to see how the survival of those microorganisms that are contaminated on those surfaces will be affected after UV light treatment. Okay? So you can see here in this chart, this is with the Listeria contamination on those coupons, especially this one is for the PVC. In the y-axis, you can see the level of Listeria, and in x-axis, we have this fertilizer. We did this for up to 7 days. Each of those bar indicates 0 day, 1 day, 3 days, and 7 days. But just fertilizer, you see there is a kind of a higher growth all the time until the day 7. In distilled water, after a day first, after 24 hours, you see a reduction in the growth, but again, there is a regrowth of those organisms on those coupons. But if you apply a UVC light, both the coupons that were with the distilled water and fertilizer, you see significant reduction after the UV light exposure. So you can see, this the same kind of a trend, same effect with another surface with vinyl. We did the same thing. We observed kind of the same data. The reduction in some cases were higher within 24 hours, and we see some of the regrowth later. But we observed kind of a similar trend with all those surfaces when you're exposed to the UV light system. So higher levels of Listeria monocytogenes, were actually found on all the coupons, maintaining fertilizer solution. If you do not do the treatment, it does maintain that one level. And then after 24 hours, the stem monocytogenesis will reduce on all the surfaces. And if you expose UV light, on those coupons, even for 1 minute, it does significantly reduce those numbers. We also have done a lot of work with the antimicrobial compounds that are extracted from natural antimicrobial compound, natural byproduct. One of the work we have done a lot is with pecan cell. Okay? We have published some of the papers with the antimicrobial compounds that are extracted from pecan cell, which is a byproduct of pecan after you sell that pecan. And we use this pecan cell in order to treat the seeds. So we use two different techniques in order to apply those pecan extract on the seeds. One is through priming, and then another one was through coating. So priming, is a low cost technology, where you allow the seed to be hydrated, in a controlled way, so they can absorb the solution and go through the first stage of germination. Okay? And that will increase the effectiveness of that seed germination also. And then during that process, you can also absorb these antimicrobial compounds inside the seeds. The coating is actually the application of external material outside the seed. If you go these days, buy seeds, for your garden, you will see a lot of seeds that are already coated also. Okay, so that is to maintain the quality of the seeds. And here, our idea was to incorporate those pecan cell extract into those coating. In our case, we use the sodium alginate, as a coating material, and then we integrated that with the pecan cell extract, and then coated the seed to see will that affect the microbial levels on the seeds if it is contaminated. So this is the overview of the process that we followed for extracting the pecan cell. We normally, I do two different ways to extract. One is the aqueous extraction, another one is the ethanol. In this process, we use the aqueous extraction, okay? And the extract were filtered and then lyophilized for application on the seeds. So we also evaluate, the germination and then the microbial, both the properties over, after the treatment on the seeds with this PSE. Those are the overview of the study that we did. For the germination, we look at the coating with the PSE and 2% sodium alginate coating. We just use the assay. Priming, the PSE for 6 hours, and hydroponics just using water, distilled water for 6 hours. But for the microbial evaluation, we did the priming at two different temperatures. One is the refrigerator temperature, and then one is at the room temperature also. So we use 3 different types of microorganisms in this study. With E. coli 157S7, Listeria monocytogenes, and all. We also use Pseudomonas species. Pseudomonas species is plant pathogens. We were also interested in looking at if the PSE actually works, minimizing risks with the plant pathogens also. And you can see over here, for the priming, we use 1:10, 1:20, and 1:30 weight by volume. Okay, so that refers to the amount of seed and times the solution, the PSE solution that you actually soak in order to do the prime. So if the seed is 10 grams, we use 100 grams, 200 grams, and 200 grams of, 300 mLs of the PSE. So similarly with the coating, those are the materials, the volume of the PSE that we use in order to treat the seeds. We also looked at the germination parameters. Will it affect the germination? So here you can see, we have this control without treatment. The PC, with 2% sodium alginate, and then coating, just the coating. The PP refers to the pecan cell extract plus the priming. Okay, so the above is the pecan cell extract plus coating, and then the hydroponic. So here you can see actually the priming or hydroponic actually maintain or increase the germination properties did not affect the germination properties. So Listeria levels on lettuces after the PSC priming and coating treatment. You can see here the control is the contaminated seed. And then hydropriming, there was a reduction, significant reduction with hydropriming alone. With PSC priming, root temperature, PSC priming refrigerated temperature, PSC coating, all of those were significant reduction in Listeria levels. In both cases, that is a priming or it was a coating. We also work on with the equal 157, and then later I will show you the slides for the Pseudomonas. You can see the same observed value that if you increase the ratio, like from 10 to 20 to 30, you see with the 30, 10 to 30, and there was a not necessarily reduction with the coating. And in case of priming, you will see much more reduction. So priming was working much more better than the coating itself. Similar trends was like observed with the Pseudomonas also. We did observe significant reduction with the coating and priming. Not much difference between the ratio, like of the seeds and then the volume of the PSE, but there was a significant reduction in both cases with the coating and then the priming. So we were also interested on looking at how we can use the byproduct, okay? And in many cases, like the rock wool that is used in hydroponic system, the rock wool has to be thrown out after you produce the hydroponic, okay? We are looking at, can we actually recycle the rock? Can we treat, like with boiling water and then or steam, and then we'll reuse the rock. Remember, like the rock wool that we use in this study, we use the fresh rock wool. We haven't like I used already grown rock wool. That is our second phase of the experiment. Okay, we'll be using those rock wools that already have a lot of rules that's incorporated. But in this study, we did evaluated hot water, especially boiling water, treatment the rock wool with the boiling water, and then treatment the rock wool with steam, like to maintain the rock wool temperature at 70 degree, 80 degree, or 90 degree. So here, this chart shows the Listeria levels in a contaminated rock wool, that was deep in boiling water, and contamination level was pretty high. We started with 8 log, but you can see, like within 10 seconds, like our treatment with boiling water, it almost went to the level of detectable limit. Okay, so here it was plateau because it is right above or below the detectable limit of the test, like that. So we achieve that within 10 seconds. And this figure shows treatment with the steam. You can see, like those 70, 80, and 90 degrees is not the steam temperature, it's the temperature of the rock wool that's maintained. Okay, so you can see at like 90, like a degree, there was a significant reduction within 10 seconds, and within 20 seconds, like the levels were almost at or below the detectable limit of the test. Okay, at 80 degree, like the reduction was achieved after 40-second treatment. Like when we start the 40-second treatment, and within like 1 minute to 5 minutes, you will see several log reduction. 70 degrees centigrade, a similar trend, like it requires at least 1 minute, like our higher treatment at lower temperature. So if you go to a higher temperature, you would get reduction much more quickly. So with this results, both the boiling water and steam can reduce significantly the microbial contamination from the rock wool. The steam treatment, the benefit of steam treatment is there will be less water absorption, because when you put in the rock wool into the boiling water, it will absorb all the water, and then you have to go through another process and remove a lot of water. Okay, and there is a promising, this promising method, but we do need to work with the actually grown and recycled rock wool, because the amount of roots that is incorporated inside the rock wool may interfere with the reduction of those microorganisms as well. We also, work, and then we're thinking about identifying other different ways other than the chemicals in order to minimize those microbial risks. And this is another experiment, we started from last couple of years. This focus on identifying and isolating the predominant microorganisms. that are present in the biofilm microflora of the nutrient film technology hydroponic system using the co-exclusion method. So in this co-exclusion study, what we did was we collected environmental samples from actually the hydroponic system. So from several different areas of hydroponic production, like a grow trays, like even from the nutrient solution, the drain area where there will be a higher number of microorganisms, and then the nutrient pump. Okay, so several locations were swabbed, and then the environmental samples were collected, and then we grow them at in our lab, and we did the full exclusion study to identify which organisms is actually predominance from the microbial flora that we actually isolated from that hydroponic system. So the swabs were taken, and then we plated in TSA, incubated 24 hours. All of those colonies, would again restrict into a new place to get an isolated colonies. And then we isolated those colonies using those isolated colonies were actually identified using the Malditov. Okay, so based upon the colony characteristics, we separated those colonies, put it in a separate TSA place, and then the Malditov was done to identify what colony was that. Okay? And then those isolated colonies were put in TSB again, and then we did the a co-exclusion study with the 1:1 ratio with all of those microorganisms. Okay. So those are the different colony characteristics, that we actually isolated from that hydroponic system. And then those organisms were identified using the Malditov. You can see several different colony characteristics. That's what we use in order to identify during our co-exclusion study. So this is the chart that shows the co-exclusion study. There were around 120 combinations. So we combine each organisms, with another organism, 1:1 ratio, and then see which will predominate. Okay. So after incubating 24 hours, we plated into TSA again to see which organisms will be left on those TSA. So here you can see this isolate 7 was the one that was identified, and then we send it to again Malditov to identify that particular microorganisms, and then it was the Pristia megatherium, which was the predominant microorganisms from all those protein isolates. So remember, we did 120 different combinations in order to identify this microorganisms. So again, after we identify that, we want to see the antimicrobial efficacy of this organism. So we use the cell-free supernatant, of the organisms against Listeria and Salmonella species. And we also determine their effectiveness in sanitizing food contact surfaces. Okay, so it's grown in the organisms was grown in TSV at 37 degrees for 24 hours. Centrifuge, filter, freeze dry. Okay, and then they have dissolved supernatant protein concentration, we determine, and then we did the antimicrobial susceptibility testing with the Gravois assay. So here you can see the Gravois assay. We did test with Listeria, E. coli, and E. coli 157 Salmonella. This is the results for the E. coli, for the Listeria. Okay, we used several different strains of Listeria. Many of those strains were isolated from the foodborne illness, outbreak. Okay, and with the Listeria, you depending upon the different strain, their zone of inhibition ranges from 11 mm to 17 mm. Remember, this includes the 6 mm of the well cell also. We also examine the zone of inhibition of the cell-filled supernatant, that organisms to all of those odors, isolates that are isolated from the hydroponic system also. So it varies, from around 9 to almost 18, mm zone of inhibition. This is the results for the Salmonella enterica, and then the E. coli 157S7. Those are on the TSA list. Okay. With the Salmonella, it is around 16 mm, and E. coli 157, we're around 15 mm of zone of inhibition. We also looked at the amino acid profiles, of the freeze dried, an EMP, because we want to see what exactly amino acids is predominant in that solution. We found that glutamate, level of glutamate was really high. And there are a lot of research that shows that glutamate contributes to antimicrobial peptide stability, surface activity, and then membrane binding, and maybe also the source for the antimicrobial activity. We use that, the extract against contaminated coupons also. Okay, so those PVC without surface and SDP, those coupons were actually used in hydroponic system. Those coupons were contaminated with the listeria, and then we actually dipped those coupons, contaminated coupons, in those extract, and then you can see the reduction. There were almost more than 2 log reduction, in each of those surfaces. This is with the Salmonella enterica. Even with the Salmonella, you can see more than 2.5 log reduction in PVC. In vinyl surface, almost 3 log reduction, and then SDP, almost 3 log reduction. So, with this study, demonstrated that the antimicrobial activity of Bacillus megatherium against Listeria monocytes, Salmonella enterica on food contact surfaces. Treatment with CFS, led to significant reduction in pathogens, up to 3 log. And then the broad spectrum efficacy combined with its natural origin and compatibility with hydroponic system highlight the potential of this AMP formulation. This technology actually offers a sustainable and safe alternative because many brewers doesn't want to use chemicals, and then isolating organisms, predominant organisms from the system, and then use their self-free extract from that organisms to inhibit the growth of potential pathogens is a huge thing for hydroponic production. So in conclusion, UVC light is an effective safety intervention. It has some limitation, but properly applying UVC light throughout the production system, and then minimizing the risk of all those inputs, including the seed, the fertilizer contact surfaces, and then the nutrient solution can actually minimize the microbial risk in hydroponic system. Hydroponic environment can enable persistent rebound over time. Okay? So higher pathogens were observed in fertilizer solution. The increase of maintaining the pH level can also be one of the ways to minimize some of the risk. But again, there are antimicrobial treatment, using UVC light or those bioactive compounds that can minimize the risk with the seed and then with the plant. Thermal treatment of the rock wool can be applied to reuse those rock wool. This study, again, we need to work on another part of the study with actually using the recycled rock wool, but we observe promising results with the steam and then both the boiling water treatment. Bacterial extract from the microorganisms, predominant microorganisms from the hydroponic system, shows strong antimicrobial potential. If we could purify, those with the microbial peptides that may have higher efficacy, and the one that we are using is more food extract that are sulfate supernatant, but it has a great potential on minimizing some of those risks with the listeria, salmonella, and then the following parts. So this is my lab group, they are my superstar, all the work that I presented today are the result of their work, okay? I'm just delivering those work, and they work very hard, I'm very much, so much proud of them, very much. So With that, thank you. I really appreciate this opportunity to present. If there's any questions, we'll be happy to answer.
Awesome. That was great. Thank you very much, Achyut. Sorry, I think there's a bit of an echo. So we actually have a question. So folks, if you have a question, feel free to put it into the Q&A section, to type it into the Q&A. We have an interesting question from Nirmal. I think referring to the work that you were talking about with UV treatment of the different materials in the hydroponic systems. And Nirmal was wondering about specifically how the inoculation and microbial enumeration was done for the surfaces if the distilled water or fertilizer was used.
So, like with the surface, we inoculate, okay? We inoculate on those coupons, and then we dip into the distilled water. So inoculation, then drying, certain time to make sure those organisms are firmly attached to the surface, and then we're dipping distilled water. When we were evaluating, we did like a consider to remove those uh, uh, loosely attached cell by running with the distilled water, and then washing through the distilled water very softly. But during the enumeration, we use glass beads in order to remove those firmly attached cells and then uh, enumerate those levels. That was a good question. That's good. Thank you. We are more interested on like cells that are firmly attached on those surfaces that was you find in a natural environment. I won't say it is natural biofilm, but it is a film biofilm that are attached firmly on the surfaces. And we use the glass beads uh, when we were dislodging those microorganisms from those different surfaces.
Cool. Awesome. That was a great question. I believe Shital raised your hand. So I think we have some other questions here. I guess. So Aaron, thank you for the question. Has a subjective panel to assess overall liking / general feedback, overall organoleptics on UVC treatment of lettuce been considered? That's a really great question. Like, so getting involved, I guess, with the sensory folks.
Yeah, exactly. We haven't done that. Like with the sensory folks, like just like watching. We did have. I didn't put the data from our colorimeter. We did have like the colorimeter that measure the color changes, but that is something that we will be probably doing like in the next phase of our experiment to actually see how this does will affect, and then using a panelist like to actually determine the differences in the effect of different doses of UV slash treatment. But that's really good, and that's on our approach. Like we are trying to do so many things. That's why you see like so many different experiments. We want to have a complete like a picture to understand is UV light actually viable. Our understanding of using a UV light, like I is not from only one surface. We want to use the UV light. If it is actually working in a hydroponic system, we want to use the UV light from different angles. That way, like if it is a surface that is not smooth, then it will get an exposure of the UV light. So, but we need to be very careful. During the production cycle, we saw, like a higher than 1 minute treatment of that intensity, 0.28 milliwatt per centimeter square, like it will affect, it will have a phytotoxic effect. So we need a higher level UV dose for fertilizer contact surfaces, but for the plant, the dose will be significantly lower. Otherwise, it will have an adverse effect. So there is still a lot of work to do in order to apply. But I am really excited because there are several different ways to do like a CIP control, like a sanitation in place, like how you do in dairy industries. Yeah, you can run the UV light, like inside the tube or inside the cyst without actually exposing. So there are different ways of that treatment. But again, that with the plant, that's our another next step.
Cool. Great questions. So before Shannon had a really awesome question, but before we get that, I believe Nirmal had a follow-up question about the previous first question, and I think Nirmal was wondering specifically if when you're drying on the coupons, that has an impact inherently on the microbial load and how significant that is, I guess, for the surfaces as well as maybe lettuce.
With the latest or with the fertilizer contact surfaces.
I think with respect to the surfaces, the contact surfaces.
So so there with the fertilizer contact surfaces, when we dry, there is a reduction in microorganisms after you inoculate and dry. So we consider the initial level, after we wash out those loosely attached cells, and then those firmly attached cells were only considered as the initial level of those contaminant on those uh, contact surfaces. Okay, so again, only those attached salts were considered, and we did dip those into distilled water and fertilizer solution separately to see how these microorganisms will their survival will be affected in this different environment. And during the illumination, we again follow the same procedure. We remove the loosely attached cells from the surface again with distilled water, run it very softly, and then we use the glass beads to dislodge the from the attached cells. So, yep, those organisms were actually attached from the attached cells, and then we did not consider the initial inoculum. The initial label was after the drying and test on the surface. We consider just before the treatment the initial. I don't know if that makes clear or not, but yeah, there is always always a good question about what is the initial label and how which you consider. And in our case, we consider the organism that were on the contact surfaces that are from the attached, and we use that just before we treat it with the distilled water or the fertilizer solution.
Cool. Yeah,
Not the inoculum.
Sorry.
Yeah, not the inoculum label. Yeah, go ahead.
Cool. Yeah, it's a great consideration, definitely. And so it's a really good question. It's something that always needs to be kept in mind for this type of work. So Shannon asked a really awesome question here. So she says that she knows in traditional gardening, companion planting, so growing different types of plants closely together intentionally, can be used to improve growing conditions. Can this potentially be applied to hydroponics to mitigate pathogen spread? That's a really interesting question.
Yeah, it can be, Sam. No, that's a really, really good idea. You just gave me some idea. So,
Yeah.
yeah, I have a nice backyard garden, and my wife, she grows different types of vegetables. And, you may have observed, when you grow different types of vegetables, together in a small plot, your vegetable is very tasty, very flavored. Okay? And there are so many different compounds, natural compounds that are produced by varieties of different vegetables that actually manage your taste also. So we do need to test in hydroponic system, but that's great idea. All right, Sanand, you just brought us some really good idea for someone to work on. And in many of those hydroponic systems, you see a limited amount of crops that's grown because you may have seen we use different types of nutrient. Okay, so the soluble nutrient, for lettuce, there are specific nutrient like that are base, and then for the tomatoes it's different, for the strawberry it's different. Okay, so if you combine all different types of crops in the same system with the same nutrient solution, you may have to compromise with some of the production yield and others, but there may be always a way to optimize those essential nutrients for each of those plants, and can be a great way to control the microbial risk in hydroponic system. But good question and good idea.
Cool. Yeah, it's a really interesting idea. And I believe Shital, you had your hand raised. So were you? I think I just hit the allow to talk. Did you have a question that you'd like to ask? Shoot.
I'm so sorry. It was an accidental hand raise.
Oh, okay. Yeah, no worries about that. Sorry, I'm still really unfamiliar with how to do these like through Zoom with the webinar. Okay, cool. Well, I had maybe one final question. If there aren't, feel free again to put in the Q&A. I had maybe one final question as we get close to ending out the hour here. Um, I think just more on a global, like a global perspective. So you've done a lot of work in these controlled agricultural environments. What do you think, in terms of food safety is going to continue to be the biggest issue as we see more and more hydroponic or controlled agricultural environment products? Like, what do you think is maybe the biggest potential challenge or problem that people haven't really thought about as much?
That's why I was like, this research work because in hydroponic system, uh, everything is connected, and like, we are actually providing nutrient water that is also very favorable condition for the growth of microorganisms. So microbiology, we know, and we talk about all environmental pathogens in stevia, monocytism is everywhere in the environment. So, in like, we didn't have any problem until now with the hydroponic system, and therefore we will have always no problem. But many of these hydroponic growers that I know, they follow really great sanitation technique. They are very proud of the hydroponic system. They are following good, tobacco training, cleaning, sanitation. Okay? That is why, if you just add one more layers of treatment, might reduce the potential risk. So, again, we don't have any much problem, but there is a potential because of this environmental pathogen can get into the system, even with the seed. If the seed is contaminated, one seed, contaminated seed can, can compromise the whole system. That is what I always get scared of. And if we follow with all these treatment procedures, with the seed, with the water, and, sanitation technique, we definitely can minimize the risk.
Cool, cool. Well, yeah, and it was really interesting work. And you you guys are doing a lot, and it's really going to continue to be an important issue as you see this sort of sector grow in produce. And so, thank you so much for your time and for sharing a really interesting research. Thank you all to the folks who attended, and also all the really awesome questions that folks had. I don't want to hold you guys up anymore, so thank you very much, Achyut, and have a great afternoon and evening, everybody. We'll see you in February. So,
Yeah, thank you so much, Mark. Really appreciate the opportunity, and thank you everyone for attending this webinar.
cool. Thank you. Have a good one, everybody.
Stay warm. Yeah.
Yeah, you too. See you.